We might say a few words about releasing factors because there's an interesting history of how they were discovered, and it also illustrates some important methodology principles that scientists use. There were two teams of researchers competing to discover releasing factors. They realized that there was something going on here in that venous supply to the anterior pituitary that was causing ACTH to be released. And so one team, I think it was in Washington State, and the other one at Baylor College of Medicine, studied the problem by going to slaughterhouses and collecting the hypothalamuses of sheep in one case and pigs in another. And then they would take hundreds of these and pool the tissue so they'd have enough releasing factor present to study. And they would put this in solution and pour it at the top of a column. And you can see the column here in the picture. It's a huge column, and it's filled with the substance. It's like sand. And if you pour material through this column, the molecules in the solution will separate out. The big molecules will get trapped first up toward the top, and the other ones will pass on through. And it turns out releasing factors are rather large. They're peptides, and they get trapped up in the upper portions of the column. You can stain them, and then as you see here, you can actually see where the chemicals are. So you have bands here that you can extract and test in other animals to see which bands have the chemical that will trigger the release of ACTH. And this is what they did. Both research teams did that, and they then analyzed these the chemicals in the appropriate band and discovered the chemistry, the peptide chemistry of these compounds. By the way, both workers got the Nobel Prize for this work.